Welcome back, everyone. My name is Lindsay Brown with the Beyond Clean team. We want to thank 3M for sponsoring this event, as well as the Competency and Credentialing Institute for their collaborative partnership. As a reminder for all of you tuned in each session, there is a resource list on the right-hand side of your screen. You'll find links, downloadable documents, all of which have been provided for you by the conference sponsor, 3M, as well as each individual speaker. So certainly take advantage of those. On the lower left-hand side of your screen is a question and answer tool. As you're listening to this presentation and all of the presentations in today's conference, feel free to interact with the speaker by submitting questions that you have about the content. Joseph Stoffel has worked for 3M for 18 years and is a microbiology specialist. His focus has primarily been in wound care, while also having supported efforts in diagnostics, sterilization, food safety, and drug delivery. Joseph leads new product development efforts in addition to refining and expanding 3M's capabilities and understanding in microbial biofilm testing. Joseph today will discuss virulence factors that make microbes more resistant to control and antimicrobial stewardship initiatives that we can take to protect patients from harm. And he does so in such an approachable way. Joseph is becoming one of my favorite speakers to listen to because he takes these really heavy topics and he boils them down in very understandable ways. So without further ado, I'd like to welcome our next speaker, Joseph Stoffel.
Welcome, everybody. I'm Joe Stoffel, microbiologist with 3M. And today we're going to talk about a really complex, but I believe a very important subject, and that's antibiotic tolerance. Today's objectives, I'm going to be defining the key terms throughout the presentation, and we're going to start with a brief history of antibiotics. Then we'll switch over and give a high-level overview of antibiotic resistance. We'll briefly touch on also how biofilms play a role in antibiotic tolerance, and then finish up the presentation talking about antimicrobial stewardship and how it really helps and how you can play a role. So let's get started. Think about antibiotics. They are a class of chemicals that specifically inhibit a metabolic process in bacteria that can keep them from growing or will cause them to die. And this is important to understand. Antibiotics are specific to treating bacteria. They do not work against fungal or viral infections. They have their own classes of treatments. When we think about antibiotic history, you may want to go directly to Alexander Fleming's discovery of penicillin by seeing that mold growing on that Petri plate. But actually, there were others that were working on this area way back in the 1880s. And it actually led to one of the first marketed products all the way back in 1910. The first systemic antibiotic that became available were sulfa drugs out of Bayer Laboratories in 1933. But for all intents and purposes, we really consider the modern age of antibiotics beginning with penicillin and its wide distribution beginning after World War II in about 1945. So when we think about antibiotics, we can break them down into 12 classes that are typically used medically today. 10 of these were discovered between 1928 and 1968, with the last class being identified way back in 1987. And when we talk about antibiotic classes, we're typically taking the compounds and grouping them based upon their structure and also their mode of action. When we talk about mode of action, we're usually referring to three basic concepts. We need to start with understanding what are the differences between mammalian cells and bacterial cells. And one of the primary differences is the presence of a cell wall. Bacteria have a cell wall, mammalian cells do not. So a lot of the early antibiotics are targeted towards inhibiting the synthesis of this cell wall. Another way antibiotics can work is that they can interfere specifically with the production of protein, RNA, and DNA within a microbe, which then can lead to either cell death or prevent them from multiplying. Additionally, other antibiotics can disrupt cell membrane function. Cell membranes typically have proteins embedded in them that are responsible for allowing certain molecules to cross the membrane in and out of the cell. If we can shut these down, again, we can prevent that microbe from continuing to live. These are some examples of maybe some of the antibiotics that you would be familiar with. The one class that I'm going to use the most in my examples today is that very first class that was discovered way back in 1928, and that's beta-lactams. We think about the discovery of antibiotics and Fleming all the way back, noticing that mold growing on that Petri plate and inhibiting the growth of the microbes that he was trying to grow. What he was really observing was something that is common in nature. Antibiotics have been produced by microbes for thousands of years. And some of the basic ideas behind why that came to be are they use these antibiotics to prevent organisms from competing for resources. So essentially, they're selecting for who they want around in their neighborhood. If there are rare resources around, they want to keep competitors out. Additionally, in nature, we see very commonly symbiosis. So they could be using these antibiotics to select specifically for other microorganisms that they want to have nearby because they may be providing them a benefit. Finally, another thought that is out there is that these antibiotic molecules are just basic signal molecules common inside the cells, and that just by chance they happen to be toxic to other bacteria. And ultimately what has happened is humans have figured out how to harness nature for medical benefit. So if we go all the way back to that first molecule, penicillin, what it has led to is to greater than 40 beta-lactam compounds today when we break them down into several groups. What I like to call the psyllin compounds, and we think about penicillin, methicillin, amoxicillin. Also, we have what we call cephums, so think of cefazolin, cephalexin, cephalosporins. And also in this group of beta-lactams are carbapenems and penems. These are typically ones that are on the drug of last resort list. Now, on the other side of the coin of antibiotics, we have antibiotic resistance. And this is when a bacteria demonstrates the ability to either prevent the entry of an antibiotic into the cell, essentially they just put up the wall and don't let it in, or they have what we call active efflux. Efflux is the ability to efficiently pump the antibiotic right back out of the cell if it made it in. And the analogy I like to give here is that you're successfully bailing the water out of the boat and keeping it afloat. Enzymatic digestion, very common, and we'll go into this a little bit more detail in a second. Bacteria also are very clever when they mutate. They can do things such as altering the target of the drug receptor site. So when the antibiotic gets into the cell, it can no longer recognize where it's supposed to go and bind to, because the bacteria may have all of a sudden tacked on a random carbon molecule or another metal ion somewhere that now has completely changed the structure of where it's supposed to go and renders the drug ineffective. And finally, bacteria have also found ways to completely synthesize new pathways around the one that is being targeted. So this is the one I kind of, instead of taking the plane, now we're going to take the train. And like I said, enzymatic digestion is very common, and this really strikes at the heart of that category that I was talking about, beta-lactams. There are a lot of organisms out there and a lot of resistance genes that code for beta-lactamase, the enzymes that can cleave that beta-lactam ring and essentially render these class of antibiotics ineffective. Now, I want you to understand that antibiotic resistance isn't something that is man-made. It is a phenomenon that is common in nature. And how we know this is, this experiment was run several years ago where some scientists went to a remote part of Alaska to an area that was largely considered to be unsoiled by man, and they took sediment samples. When they carbon dated these sediment samples, they were greater than 30,000 years old, and they were also able to culture microorganisms from these sediments. And lo and behold, the organisms that they grew up from these sediments contain antibiotic resistance genes. Now, how can that be? So we think about that first statement they made, like, why are they producing antibiotics in the first place? And one of the things that was considered is maybe they're just trying to protect their area and not have to compete for those resources. But this is what I would consider all is fair in love and war. If one organism is developing a weapon to keep you out, it's only natural that someone is going to try and find a way around it. And that's what we have, is bacteria have evolved ways to overcome what we call these selective pressures. A selective pressure means that when you're putting pressure on a microorganism, the ones that are able to adapt and get around it will survive, while the ones that aren't are selected out and they will die. So we also want to consider that everything in nature lives in a state of balance. So it is logical to assume that organisms that produce the antibiotics are also going to have a mechanism on how to control the production of that antibiotic. The example I like to give here is how, like, we produce insulin. So when our blood glucose gets too high, our body pumps out insulin to regulate how much is actually there. So it's logical to think that organisms are doing the same thing, that if an antibiotic concentration gets too high, it has now another gene that codes for how to stop or control that antibiotic. And then this now starts to fall into the category of, even though these organisms are fighting each other, they also like to share. So these genes are getting out of these organisms and into other organisms. And one way that that is potentially happening is that these organisms are routinely growing in nature. So when they produce too much material, even DNA, and it becomes too ubiquitous inside the cell, sometimes they can just excrete it out into the environment. Additionally, cells are multiplying and dying all the time. And when they die, they can just burst open. And now all their genetic material is just free and available in the environment. And if the one bug is in the right place at the right time and happens to acquire up this free genetic material, DNA, that's free floating out there, this is how they're able to acquire these genes. And the other thing, whether we realize it or not, is that bacteria do have sex. They are constantly sharing genetic material amongst each other all the time. Another mechanism that bacteria have evolved to help withstand those pressures that they routinely come across in nature is what we would call a biofilm. Biofilms are the state in which microbes want to reside in nature. They don't want to be free-floating organisms in a test tube that we typically see in the laboratory. They want to be adhered to surfaces, and they want to cover themselves in this slime matrix that we call the extrapolymeric substance. And it's this extrapolymeric substance that makes them a lot more tolerant of the antibiotics. So if we consider this cross-section of a biofilm, how are they able to withstand these antibiotics? One is that extrapolymeric substance can slow or completely stop the penetration of the treatment from getting deep into the biofilm, offering protection to the cells that are at the bottom of that biofilm. Additionally, bacteria have also evolved a way to hibernate, essentially become metabolically inactive, and we call these cells persister cells. So even if the antibiotic was able to penetrate down into the biofilm, the ones that are existing in this persister state can just completely ignore the antibiotic existence. So now we take a biofilm, mechanism, an antibiotic resistant mechanism, and this is how we end up with the term superbugs. So the common definition of how we would call out superbugs is when a particular strain of bacteria is resistant to the antibiotics that we would typically use to treat the disease that they're causing. And another key tenet to a superbug is that they're going to be multi-drug resistant. So when we talk about multi-drug resistant, there are a couple different flavors that we can take. Again, let's consider MRSA. So an MRSA may not only be resistant to methicillin, which is in the title of the organism, but it may be resistant to a whole bunch of other beta-lactams in that class. So it also may have resistance to the carbapenems and to the cephalosporins. Now, we also have Staph aureus that are resistant to other classes of antibiotics like vancomycin-resistant Staph aureus. Vancomycin is a glycopeptide. And so it wasn't really that far of a stretch to think that eventually, someplace down the road, an organism was able to combine both of these resistant organisms, and we do find on occasion methicillin and vancomycin-resistant Staph aureus. Now, I've also listed out here really common organisms that we see routinely in nosocomial infections and outbreaks in communities, because they are very capable of acquiring a lot of these genes in nature and sharing them amongst each other, as well as containing a high level of pathogenicity. Now, the one organism that maybe you remember before SARS-CoV-2 outbreak happened here with the pandemic was Candida auris. This is the one we were really concerned about a year ago. And the reason why we are concerned about this organism is that it is a fungal pathogen. Now remember, antibiotics are ineffective versus fungi. And the number of antifungal compounds that we have available to us to treat medical infections is significantly smaller. There are very few good antifungals that aren't toxic to humans for treating these. So now when you come along with an organism that becomes multi-drug resistant to a very small set of drugs that are available in the first place, this definitely becomes concerning. So now we're going to switch gears a little bit and talk about antibiotic stewardship. When we think about it from the hospital or the clinic standpoint, this is how the CDC would define it: the effort to measure and improve how antibiotics are prescribed by clinicians and used by their patients. And they've published these 7 core elements that they want hospitals and clinicians to implement to help prevent unnecessary use of antibiotics. And a lot of these are pretty self-explainable. You need good leadership to bring in and implement these, and then you need accountability to make sure people are using the elements. The 2 things that I really believe are important here is education and expertise, because it can go a long way that if we can identify specifically what the infections are early and apply the appropriate antibiotic earlier, we can prevent some of this unnecessary use and inappropriate use of other antibiotics, which then helps propagate more resistance. But in reality, when we talk about stewardship, it goes on way further than just hospital and clinical use. So we look back in 2011, 50% by weight of all the antibiotics that were consumed on Earth were given to animals. And they weren't even given to sick animals. The majority was given for the idea of growth promotion. Now, largely this has kind of been debunked lately, that it actually giving antibiotics to animals doesn't make them grow faster. What it does is it just prevents more animals from getting sick. So at the end of your stream, your production stream, you've had to cull out less animals that got sick along the way, and your production goes up. Now, why this is such a huge issue as we think about farm ecology is the perfect environment for spreading these resistance genes. Now, we think about like basic hygiene. Why do we wash our hands? Why do we try to stay distant? Is we want to make sure that we're preventing the fecal oral transmission. So that's typically how we get sick a lot of the ways. That's why, like I said, that's why we wash our hands. And you think about on a farm, you have animals, they're depositing their feces, then they're continuing to live in those feces. Their pen mates live in the same feces. So it becomes really the perfect environment that if one animal begins to develop a strain of resistant bacteria, that they're going to spread it to everybody else in that farm eventually. And we also need to take in consideration of the farmers who care for these animals and how they become susceptible and exposed to these organisms as well. Now, we have seen bans of this type of antibiotic use in Europe and reductions in the US. So any antibiotic that is primarily for human use can no longer be used this way in agricultural, but we do still see a problem of this unnecessary use of antibiotics. They really should just be used for treating the sick animals. Another thing that we encounter is that in a lot of developing countries around the world, antibiotics are available over the counter. So we can get a lot of unnecessary use, because if someone just feels ill, they don't quite understand what they might have, and they run down to the pharmacy and pick up a bottle of tetracycline, and it may not be the appropriate prescription that they need. And also, we just have to deal with basic human nature. You know, we get sick, patients go to the doctor, we want something to make us feel better, and a lot of times patients will demand an antibiotic prescription. And clinicians, even to the best of their intentions, they want to help their patients. They want to make them feel better, put their minds at ease. And even though they've tried to convince them that you're probably just dealing with a viral infection and your body will clear it itself, they still want to help, and they may give them that antibiotic unnecessarily. So why is stewardship so important? Well, the obvious one is, I think, if you go way back to that earlier slide, is that we are running out of options. No new classes of antibiotics developed for human medical use since the late 1980s. But I also want to just give a few stories of why we need to try and do better. So you may have come across this resistance gene in the news because it's very common in nosocomial outbreaks with organisms like Klebsiella and Pseudomonas containing the gene NDM1, which stands for New Delhi Metallobeta-lactamase 1. So again, you typically see this again, makes the bugs resistant to lactam, beta-lactam class of organisms. But why this is also is important is that routinely what we find is that if an organism has acquired the NDM1 gene, it also contains several other resistance genes as well. And why this is important is because these genes can spread and go global quite quickly. When the NDM1 gene was first discovered, largely in India and Pakistan, within 5 years of its discovery, it was being isolated on every major continent on Earth. We also need to consider that antibiotic usage can trigger other diseases. And the most common example that I could think of was C. diff. So C. difficile, Clostridium difficile, is a spore-forming organism that normally just resides in our gut. And it just hangs out and doesn't do anything, because our normal flora keeps it suppressed. Now, if we need to take an antibiotic, or if we unnecessarily take an antibiotic, knock down our normal gut flora, and then when we're done taking that antibiotic, this is when C. diff sees an opportunity to come out of those spores and cause serious disease. So the main thing is, that's why they tell you to take the probiotics and try and keep your normal flora, is to kind of keep organisms like this suppressed. And C. diff is one of those organisms that we find routinely to be multi-drug resistant. Another concept that we really need to consider is all the diseases that we take for granted as just being generally curable today. And the example that I like to give here is strep throat. I've had strep throat, my kids have had strep throat, and I'm sure we've all dealt with this routinely. And when it comes, we don't think too much about it. We take our kids to the doctor, we get the test, we get our prescription, we go home, and it's gone within a few days. However, just over 100 years ago, at the turn of the century, the 1800s into the 1900s, the childhood fatality rate for a kid that would develop strep throat was 80 to 90%. That's pretty astounding when you think about something that we take for granted today as strep throat. 80 to 90% fatality rate 100 years ago before antibiotics. So I like to listen to a podcast, and I'll give a shameless plug for This Week in Microbiology. And one of the podcasters, one of the contributors on the podcast is Dr. Michael Schmidt. And then in a podcast he had given talking about antibiotic resistance, he kind of threw out this idea, which really struck home with me. It's antibiotics have enabled the expansion of modern medicine. Well, what did he mean? Well, we think about all the really complex surgeries that happen today. Open heart surgeries, dirty surgeries on stomachs and intestines. These are the types of surgeries that really make us susceptible to infections. And without antibiotics, these surgeries would really struggle to have positive outcomes. It wouldn't be because the surgery itself was not successful. It would be the onset of the coming infection from having to be cut open for so long. And we also have to consider organ transplants and cancer treatments. These are treatments that leave people immunosuppressed for long periods of time. And without antibiotics, these people would have to live in a bubble because they are so susceptible to infections. And the strange thing about this is when we become so immunosuppressed is that we open ourselves up to being infected with organisms that are typically don't infect humans. So it is really important. Some other concepts that are being studied out there and where some early links are being drawn as we think about all the work that has gone into studying our gut microbiome and our surface microbiome on our skin, and how important it is to keep a normal microbiome intact. Because there is data that suggests that when we see microbiomes that are severely altered, that they have some correlations with the development of asthma, allergies, obesity, and adult onset autoimmune diseases. So this is an example that I like to kind of walk through to really drive home the point of how important stewardship is. So in the 1940s, World War II soldiers, venereal disease was a rampant problem. Gonorrhea was pretty common. But penicillin then comes along and it's a wonder drug. You can get cured of your gonorrhea pretty quickly with just 72 milligrams of penicillin. By the 1960s, now you needed 40 times more penicillin. Plus, you needed to take another medication that kept the penicillin in your system longer because our body wants to get rid of the penicillin pretty quickly. So now you need 40 times more and an additional medication just to get rid of the same gonococcus. And by the 1970s, gonococci have now developed complete resistance to penicillin. That only took 30 years. So we could say that we're still living in the golden age of antimicrobials and antibiotics. I do believe that for the most part, but you do have to wonder for how much longer. And that's why we all play a role. So let's think about what's out there on the horizon. So, and this is not only true for antibiotic discovery, it's really true for any drug discovery, that from rock turning and early discovery all the way to bringing that drug to market, latest estimate about $750 million to develop that drug. And that's kind of an old number, so it's probably even more. So especially in the antibiotic realm, it's not a real good business proposition because when we think about going to purchase antibiotics, I don't think there's any one of us that are going to be willing to pay $100 or $500 per pill for a novel antibiotic, like we would maybe for a cancer treatment. So a lot of the rock turning and early discovery is left to startups, and then hopefully these startups can push these programs along far enough and show really good promise, and then hopefully be picked up by a big pharma company who can then take it across the finish line. But with a lack of a new class of discovery or other just basic antibiotics, what are we left with? And one of the best strategies that we have to date is to cocktail the drug. So I think you see this pretty commonly today is that when you get an antibiotic, it has a brand name on it, and it may be a combination of several different antibiotics in one pill. Additionally, there are a lot of what we call resistance mechanism inhibitors. So like we can pick on the beta-lactams again. So now we're seeing antibiotics used in conjunctions with other drugs that inhibit that beta-lactamase enzyme. There's also some really cool technology that's out there that is kind of like what they call a Trojan horse technology. So bacteria are routinely looking for rare elements and those kind of key nutrients that they need to take into the cell. So what we can do is we can take something like iron and tag it onto an antibiotic, trick the cell into taking it inside. The cell will cleave off that iron moiety, and then hopefully now we have an active antibiotic that's made it into the cell and then can do its work. I think one of the things that is probably one of our best strategies is that even though the rate of new development of antibiotics has slowed down, we are developing antibiotics within the existing classes. And what a lot of these do, they end up in the category of what we call drugs of last resort. So these are the ones that we keep on the shelf. We keep them in reserve for when all else has failed. But these typically have issues associated with them, and that's why we don't see them in wide use. They typically tend to have a cost associated with them, so that could be a socioeconomic issue. They're not always widely available. They may not be at the hospital that you need them to be. And unfortunately, a lot of these have severe side effects or can lead to permanent organ toxicity. So this is kind of like my final thoughts to just kind of walk you through about antibiotic resistance, antibiotics, and stewardship and why it's so important. So I'm going to ask you the basic question. Would you pay $1,000 for a $1 candy bar? And I think we would all pretty much agree and say no way. However, what if your life depended on it? And that's what I'm talking about when we see bacteria and when they're acquiring and developing new antibiotic resistant mechanisms. It is very expensive for these cells to run these antibiotic resistant mechanisms because it's above their normal state. They have to expend more energy, they have to use more resources, so it's just not a state that they want to exist in. So if we can cut down on our unnecessary use of antibiotics, microbes are not going to be pressured to use, acquire, or develop new mechanisms of resistance. And additionally, going back to that education piece is that if we can do better on targeted therapies, using the right drug at the right time, and then us as the patient follow the prescribed regime, and this will be my plug for if you ever get an antibiotic and it says take 2 pills per day for 10 days, but you start to feel better on day 4, please take the antibiotic every day for 10 days. And this is why. Because that antibiotic coupled with our host immune system will effectively clear every organism from the infection. So even if by chance you develop a resistant mutant in your infection, that antibiotic is going to take care of the majority of the organisms there, and then your host immune system, your macrophage, are going to come along and clear out the rest. And we will not have new resistant mechanisms or resistant organisms out there. So I want you to realize that less than 1% of bacteria are pathogenic. Let's do our best and let's not aid the noisy few. Thank you for listening.
All right, Joseph, thank you so much for sharing this information. Great advice on using antibiotics in our personal lives as well. We do have a couple of questions that came through for you, and a reminder to everyone who's tuned in today, if whatever stuck out to you in this presentation, if you have additional questions, feel free to submit them in the question and answer tool on the lower left-hand side of your screen. Joseph, to get the Q&A session started, I'm going to start by asking, can you share some insight into ways that technicians in the field can promote antimicrobial stewardship when performing reprocessing tasks of medical devices?
Sure. Excuse me. Sure. So I think it all really boils down to making sure it's the appropriate process at the right time. Now, I will admit I'm not an expert in device reprocessing and terribly familiar with the field, but I think there's some basic ideas that we can consider is when those instruments are coming back, and they're going to be soiled, you're going to have, processes, protocols in place to use to clean those instruments. And if we don't think it's appropriate to use, say, like, high-level disinfection, although that probably is something that's just automatically built into the protocol, it's all about like, is using the antimicrobial, is using the high level disinfection appropriate at the right time? And really kind of leave it up to you guys, the experts in the field, to consider when it is appropriate. But I understand as well that, there just may be protocols in place and you just need to do your job.
All right. When thinking about superbugs and their microbial resistance outside of the bacterial family, what are some concerns you have with novel and emergent diseases?
Well, I think one thing we see, especially if we're going to use, the current pandemic, SARS-CoV-2, the virus, and then it's the disease that it causes, COVID-19, is that what we're finding largely that it's not necessarily the virus that is what is killing us, but it is putting our bodies into a state that makes us susceptible to other things that come along the line. The one you're seeing a lot are bacterial pneumonias. So you think about now, pneumonias are typically pretty hard to treat depending on the patient. And now if the pneumonia onset is an antibiotic resistant organism, again, now that kind of just feeds into what are the drugs that we are going to need to use. The patient is already probably in a pretty bad state because of the cytokine storm that may be occurring. They may be ventilated, so that's probably the best example, the most relevant example I can give. But also additionally, when we think about our, cancer treatments, organ transplants, these people that are really immunocompromised, the really interesting thing is that a lot of times they get infected with non-pathogenic organisms, things that we encounter every day in our life. We just shrug them off, but these people become susceptible. So to what may not be a superbug to you and I may be a superbug to somebody else. So it's just really something important to consider.
Okay. This is a clarification question. If bacteria can reproduce by having sex, then why do they have a binary fission process? Can you kind of give a little bit of background on the difference there?
Sure. And I guess we're using the word sex maybe a little too loosely. You're absolutely right. I mean, it is asexual reproduction, binary fission. The cells just kind of like replicate their DNA, divide it in two halves, elongate and split. But there is an organelle, and that's not really an organelle because it's bacteria, but there is a structure that bacteria will produce, and it's called the sex pilus. Essentially, it is a tube that bacteria can protrude from the cell wall and then essentially hook up with another bacteria, and then through that sex pilus transfer plasmids and other genetic information. So it's more of like, because of, the anatomy of it and the fact that we're exchanging genetic material, that it's more of like a sex act rather than necessarily, sex in the general sense for reproduction.
Okay. Thank you for that. This next question says, I heard you talk about a podcast you're listening to. Could you share more about that?
Yeah, and these have really become a fantastic suite of podcasts, and I would recommend them to anybody ever since a friend of mine, a fellow microbiologist, turned me on to them. And it's they're called This Week In, and there's a This Week in Microbiology, This Week in Virology, Parasitology, Immunology. There's a whole suite of them, and they have essentially just a group of virologists, microbiologists, experts in the field that just kind of get together, and the way they sort of classify it as it's just buddies getting together and talking about science. And the thing that I really appreciate the way that they deliver it, and kind of the proof of it is that the This Week in Virology podcast, since the beginning of the pandemic, has just simply exploded in listenership. And not just by virologists or just, even biologists in general, but by lay people who just want to know more about what's going on. And why it's so successful is that they do a fantastic job of communicating that really deep scientific information to lay people. So it's really understandable, relatable. So I would start there. And there's other ones out there. Elio Schechter has a kind of a blog called Small Things Considered, that he does an excellent job with a group of people that just talk about microbiology in general. And I will say that this isn't just, disease, healthcare, microbiology. It is just microbiology in general. But I would still encourage you to kind of check it out because, I talked about in the presentation that in biology, we largely just talk about balance. We want to live in balance, microbes, and everything around us, we want to live in balance. So if you can gain some understanding from, say, one podcast that is just talking about an organism that is a bioluminescent organism that lives in a squid, and then how that may relate eventually some other podcast down the road to a different organism and some mechanism that it has that cause disease, it really becomes kind of enlightening and just gives you a great appreciation for the world around us, and then really kind of help you to weed through, the massive information that we get bombarded with today, depending on where you want to land, the feeling that there may be a war on science. It can help arm you to be a very good person to discern through what may be noise and what is valid science. So I would encourage you to start there. And then by kind of like using that as your gateway into it, then it opens the world to a lot of other resources that come along that help you find additional things. Additionally, like some of the professors or people that are on these podcasts offer up, like virology or microbiology or immunology 101 courses that are just free available on YouTube.
Okay, wonderful. That actually is a great lead-in to this next question that says, me and a couple of my coworkers are really interested in microbiology and how the concepts of it affect our job in sterile processing. What advice would you give us to learn more and stay engaged with updates in this field?
Yeah, it can be daunting, but once you practice, you can get pretty good at it. It would be to seek out and read the journal articles. What is the latest research? And I remember, when I started out in this field, it wasn't something that I was really apt in wanting to do because it requires time. But now in the digital age, a lot of these things like podcasts, YouTube videos, there's a lot of information out there that, distill it down that make it a lot more relatable, a lot more understandable. And I think if you can just kind of get the activation energy to start, you could become hooked to want to know more. And then you can start to connect dots, the more you listen or the more you read or find out about it. It's just like, oh, I learned about this, a month ago, and now that relates to this, and now the dots come together. And it may actually have an effect on how you approach your job as well. It does for me routinely when I go back into the lab and how I might want to design or approach an experiment. Some of the things that were discussed from a completely different related field in microbiology, but there was a methodology or a purpose behind it that was relatable that now I can apply. And, I would encourage everybody to maybe have an open mind that it may not be obvious at the start that it really applies, but I think, if you practice and you get enough information, you'd be really surprised at how relevant it is.
Okay, wonderful. Do you write articles in any publications that we can check out?
I do. I'm not, since I work for a company and I'm not an academic, so publications are not my priority. It's products, and delivering from my employer. But I do have a couple publications out there. A recent one is on wound repair and regeneration, where I talk about different models that we use for testing antimicrobial products. And as Lindsay mentioned in the beginning, I have a primary focus in wound care. So that paper does have a wound care focus where I'm looking at topical antimicrobial wound care products. But a lot of the tenets in that paper are sort of to describe, what is the appropriate way to test when you're developing or evaluating maybe to see what is the best product out there available. And from like a device reprocessing side, even though some of the models that may be in that paper aren't completely relatable to, say, high-level disinfection, but the concepts in that paper that I outline and a lot of the reference that I use do talk about those types of models that you can then kind of link to. So yeah.
Okay, this next question says, who are some current influencers in microbiology or in the microbiology space that we can learn about?
Oh, that's a really good question, and I'm totally gonna blow it because I'm terrible with names. But if I'm gonna kind of punt on this question, and again, I will give the plug for This Week in Microbiology. So that's it's hosted by a virologist, Vincent Racaniello, and then he has a series of guests. And two of the people that are routine on there is the one I mentioned that has his own blog, Elio Schechter, who is, I believe he's in his 90s now, but just one of the most fascinating people that you would ever listen to. And just because of the wealth of the knowledge. And then Michael Schmidt. But more importantly is that, again, it becomes a gateway. If these people are really well connected, they know the names of the people kind of like in the spaces that if you go in and you Google This Week in Microbiology and any topic that you're interested in, I guarantee you there's going to be a podcast that will pop up. And then if you listen to it, you're going to get names and you're going to find the people. And then you can search out their articles, find out about their research, what universities or institutions they're working at. Again, like in this digital age, the availability to get to the information is amazing. It's not the card catalog that I grew up with.
All right, wonderful. This next question, why did you get into this field and what inspires you? I think a lot of people are just curious about microbiology in general in this group of attendees. This is wonderful. But why did you get into the field? What inspires you about it?
And this is great. I'm glad I got this question because I'm always jealous of when I listen to those podcasts and they have guests on and they get to talk about their history. So this is, I guess this is my one opportunity. Yeah. So I went to the University of Wisconsin La Crosse, and I knew when I was like a freshman in high school that I was going to do something in science, and I took that basic career education class we all have to take, and I was flipping through a catalog, and there was a course or a profession, nuclear medical technologist, as a kid from a small town, that just sounded phenomenal. It's like, that's what I wanted to do. And I made up my mind, and that's what I was going to do, and just focused on getting there. And then I got to college, and then I realized I didn't like chemistry so much. And it was a profession that was largely based on chemistry. So like, I think most of us who have gone to college, you kind of get there with the intention of doing one profession, and then we end up taking left or right turn. And a friend of mine was taking a bacteriology course, and I'm like, that sounds interesting. So I took it with her, and then I was hooked. And that was it. I knew just there, I love the lab work. I love the concepts about microbiology. And then just every course that I took after that just made me love it even more and more. And then unfortunately, when I graduated, there just really weren't a lot of good microbiology jobs out there without getting a higher degree. And I'm not an academic. I'm a farm kid who just loves to work. So I wanted to find a job, and that's where I got my kind of first job in pharmaceutical development. But eventually made my way back into microbiology at 3M. And then, and then with the healthcare focus, I think that really kind of helped me kind of love it even a little bit more, because now it's not only a field that I really love doing, but it also has the slant of I'm doing it to help people, to maybe save people's lives or to minimally just make their quality of life better. And again, like, in this current age of with COVID-19 and SARS, I think we will see an explosion of people really interested in becoming virologists and microbiologists. And I think for everything that I talked about from that microbial stewardship slides on, really highlights why it's important that we need people working in these fields, understanding these fields, not only just like the people who are going to discover the next round of antibiotics and antimicrobials, but just people, that work with them, that are the consumers, the customers that use these, and make sure that we're using them appropriately. Ask the questions of like, is this the right way to do it? Is there a better way to do it? And having that background really can be valuable in helping you make those decisions.
Thank you for sharing that background. As a fellow farm kid, I appreciate that. All right, so this next question, a bit loaded, but let's go for it. Is the COVID-19 out of control, or is it out of range because there's still no vaccination developed?
Oh, man. Okay, so I'll do my best. I think the major problem with, especially if we're going to talk about, the United States where we live, is other countries had the ability to enact national plans, and really be consistent with the messaging, had leadership that would, and then largely then have populations that would kind of, hopefully for the majority of them, fall in line and do the appropriate recommendations that kept coming out. And we don't see that here. And I think that's part of the problem, is that, it kind of goes back to that. I said there's a lot of information out there. It's being able to weed through, like, fact versus fiction. Who are the experts that you can trust? Who qualifies as an expert? And I think, from the vaccine side, I think it's going appropriately. I think, you want to trust the people who work in the field and understand it. And it sounds like, from all intents and purposes, that, the FDA, at least from the United States side, that all the precautions are in place, all the protocols are being followed. It's just that we are moving at a really rapid pace, that we don't typically work at. So it does feel rushed, but I think from all that I have gathered, and they talk about this relentlessly on that podcast, again, I keep talking about This Week in Virology, that the safeguards are in place. They're doing all the right things. There have been a couple of studies that have been stopped because of one adverse event, and that's exactly what you do. So I think when the data comes out and the vaccines are shown to be safe and effective, that everyone should get one.
Okay. Joseph, you've brought up the podcast multiple times. I'm going to put you on the spot and ask a favor of you. Will you send me a list of blogs and podcasts that you think would be beneficial to post in the resources list? And I can do that for all the attendees that are watching today, and also that watch this session on demand later on as well.
Absolutely.
Awesome.
Be my pleasure.
Awesome. So anyone tuned in right now, you can check back in the next couple of days to the same link that you used to access the session and find those resources in the resources list. Thank you so much for that. I think that will be a great help. And also a great lead into the next question that says, can you share a little bit about career pathways for microbiology related to surgical instruments?
Oh, man, that's a tough one. I'm kind of use my experience working for a materials company, a healthcare company, about, what we do is that when it goes into, say, developing new products, new devices, there are a lot of roles that microbiology, or a lot of ways microbiology play a role. So it may be the investigating the intent of the use of the device. Does the device itself have something to do with treating an infection? There's microbiologists that are needed to do that types of testing. A lot of devices need to be sterilized. So sterilization, there's microbiologists that are required to do the sterilization verifications, and also then just, like the biological indicators, the methods of sterilization, you know, if it's heat, radiation, ethylene oxide, X-ray. There's microbiologists that are needed to work in that arena as well. And there's microbiologists in quality control that need to be able to create the documents, write the protocols, that understand the science to make sure that they're done appropriately. Yeah, I think that's about as deep as I can go.
All right. Well, thank you so much for sharing that information. That completes the list of questions that came through. Joseph, thank you for an engaging and enlightening presentation, sharing some really in-depth information and making it approachable. As always, we certainly appreciate it. Yes, and just a reminder for everyone who is tuned in, there is a 15-minute break between each session. You'll get an extra 5 minutes. So if you haven't grabbed lunch yet and it's lunchtime, actually, let me see if this one question in antibiotics. Okay, I do have one additional question that came through. You thought you were off the hook, but
fire away.
Let's see. In antibiotics resistance bacteria, which one is the harsh one? Does that question make sense?
So maybe what's the worst?
I think so. Yes. Yep.
Yeah. You know, and this is where it gets complicated for microbiology and you know, and pathogenesis in general. Is typically it's not the same for everybody. I'm going to probably get it wrong, but I'm trying to think of the equation. Is that the state of the disease depends on the virulence of the organism times its density divided by the host response. So if you have a really robust immune system, for the most part, a lot of us may not have a real bad interaction with those pathogens, but there's some of us that are a little bit less, that may have a little bit more of a problem. And then you multiply that again, like what is the organism? So it breaks down like gram positives, gram negatives. Interestingly, you know, gram positives have a really thick cell wall, but in general, it's easier to get antibiotics across that membrane, where gram negatives have a thin cell wall, but they kind of have a double layered membrane. So even though it's thinner, it becomes harder to get antibiotics across. So when we think about gram-negative infections, you know, Klebsiellas, Pseudomonas, Acinetobacters, those are the ones you really see a lot of times in nosocomial outbreaks. You know, and it's because there's fewer antibiotics that work against them. Pseudomonas is just ubiquitous in nature. I don't know if a lot of people realize, but Pseudomonas can eat triclosan. So, you know, they're really tough bugs. Pseudomonas, I guess, if I had to pick one, probably would maybe be the answer to that question, because it's a soil organism. It's everywhere. You're never going to get away from Pseudomonas, and it really has just found ways to really exist and kind of, I don't want to use the word dominate, but I mean, when you get it in a wound, it opens up the ability for other microbes to move in. It becomes really hard to cure. Pseudomonas is a huge problem in burns. Cystic fibrosis patients, it's usually that Pseudomonas pneumonia that they end up with that really sets them back a long way. But, you know, there are other organisms that produce a lot of toxins, you know, like Clostridium, you know, they produce a lot of toxins. You know, there's certain foodborne organisms, Shigella, Legionella, Listeria, they produce a lot of toxins. They can have extended spectrum beta-lactamase resistant profiles as well. So it's a long list. I don't think I could just pick one. It really depends on the niche, when, where, and who.
Okay, wonderful. Glad we were able to sneak in that last question. Thank you, everyone, for tuning in today. Thank you, Joseph, for sharing that information with us. Like I was saying before, there will be a 15-minute break before the next session. So if it's your lunchtime, wherever in the world you exist, or dinner time or midnight snack time, whatever it may be, grab a snack, assemble a tray, do a jumping jack, whatever you need to do before joining us again in 15 minutes. We're so glad you're here. We're so glad you're here, and we'll see you in the next session.